The topic of this mini lecture is epithelial tissue, specifically recognizing and naming the different types of epithelium. This video will help you recognize what you're looking at when you go into lab and examine different kinds of epithelium. There are two functional categories of epithelium: glandular epithelium and the covering and lining epithelium. Glandular epithelium refers to epithelial cells that have clustered together to make a gland, an organ that secretes a particular substance, like a sweat gland or a sebaceous gland, which secretes the oils on your skin. Glandular epithelium also makes up the endocrine glands, which you'll hear a lot more about in 242. In this video, we're going to focus on the covering and lining epithelium. One of the key functions of epithelial tissues is to provide a barrier. Epithelium separates your internal structures from the outside environment and separates internal structures from each other. So what you're looking at here is a line of epithelial cells going across the screen. These are actually from the small intestine. When you see a sheet of epithelial cells, one side of the epithelium is free, and the other side of the epithelium is attached. The free surface is called the apical surface, and the attached surface is called the basal surface, or sometimes you'll hear the term basolateral. The basolateral surface sits on a sheet-like structure along there called the basement membrane, or basal lamina. If the epithelium is lining a hollow organ, like a blood vessel or the digestive tract, the apical surface is the side of the epithelium that faces into the hollow interior of the organ. This hollow center is called the lumen. In the living body, this space might be filled with fluid, like blood. In the digestive tract, it's filled with digested food. But under the microscope, the lumen is just empty space. If you're looking at skin, there's no lumen, but the apical, remember the free surface, is the surface that faces the outside, the superficial surface. So, when you're looking at epithelium under the microscope, you want to first figure out where the lumen is. What you're looking at here is an artery on a slide. With the artery, it's easy to find the lumen because the artery is small enough that you could put the whole cross-section on a single slide. So all you need to do is look for the big empty space, and that's here. This is the lumen, which would have blood in it in living tissue. And your epithelium now is going to be around the lumen. See if we can increase in magnification and show you some of that epithelium. So there's our epithelium. It's very thin, but you can see a layer of cells along the surface. You can even see some blood cells actually in the lumen. It may not always be easy to figure out where the lumen is. For example, if we look at a piece of small intestine, the organ itself is too large to put the entire cross-section on a single slide. So here, you're just looking at a piece of the cross-section. And if you notice, there's empty space on both sides of the piece. There's empty space over here, and there's empty space over here. So which side is the lumen? Here's where it helps to have some idea of what epithelium is supposed to look like. And this is where you look at the figures in your text or your lab manual for reference. So this is what the text says we should be seeing. We're in the intestine, so we're looking for simple columnar. We should see vaguely column-shaped cells with a clear nucleus. Okay, so let's take a look at both sides of our sample, of our small intestine. You can't see much at this low magnification, so I'm going to increase in magnification and take another look. Make sure to focus, or you can't see anything. Increase in magnification. You're still looking for the edge. I'm not really seeing a defined layer of cells like I should see if I'm looking for epithelium. So let's take a look at the other side, see if that looks more like what we want. Now, the intestine has a lot of folds to it, so it's not always as straightforward as you might wish. And what you see under the microscope is never going to look exactly like it does in the book. But this looks a lot more like the columnar in the book than the other side does. You can kind of see column-shaped cell there, another one there, there. This is the epithelial side of your section. So remember, look for the lumen. The lumen's over here. The epithelium will be around that. And when in doubt, compare what you see under the microscope to the images in your text. Okay, so now that we've talked a bit about where to find epithelium, let's talk about the different types of epithelium. Epithelium is classified in two ways. First, by the number of layers of cells, and second, by the shape of the cells in the epithelium. If you take a look at the number of layers of cells in the epithelium, you have simple epithelium, in which you just have a single layer of cells. Or you can have stratified epithelium. In stratified epithelium, you have multiple layers of cells stacked up on top of each other, kind of like bricks in a wall. Now let's see what that looks like under the microscope. Here's our simple epithelium. This is in the lung. So in the lung, you've got some simple epithelial cells there. In the lung, you have a lot of lumens. Here's the lumen. Here's another lumen. And remember, the epithelium always lines the lumen. These are actually the air sacs that fill with air when you inhale. And each lumen has a lining of simple epithelium. It's actually a simple squamous epithelium, very flat cells, so that you can have exchange of oxygen and carbon dioxide. Here, we're looking at the esophagus. And in the esophagus, you have stratified epithelium, so multiple layers of cells, stratified squamous to be specific. Our lumen is up here, our empty space, and there are multiple layers of epithelial cells stacked on top of each other to protect your inner tissues from any rough-edged, half-chewed food you swallow. So here's our apical surface, and all the way down here, you've got the basal surface and the basement membrane. And in between, you've got layer, I'm not even going to try to count them, but you've got layer after layer of cells. So epithelium can also be classified according to the shape of cells. Squamous cells are what I was showing you before. These are flattened cells. They look kind of like a tile from the top and kind of like a fried egg from the side. If squamous epithelium is the fried egg, then the nucleus of the cell is the yolk. Cuboidal cells are shaped like a cube. They're about as wide as they are tall. And the nucleus sits right in the center of the cell. The third shape you can have are columnar cells. Columnar cells are, of course, shaped like columns. They're taller than they are wide. Sometimes it's not easy to tell if you're looking at cuboidal or columnar, so look for the nucleus. In columnar epithelium, the nucleus often sits closer to the basal side of the cell, whereas in cuboidal, it's usually in the center. In stratified epithelium, the epithelium is named according to the shape of the apical layer of cells. Let's look at some examples. Here we're looking at simple squamous epithelium. I already showed you simple squamous from the side. Here it is again, another view of lung tissue. So you can see the flattened cells there. This other image shows how simple squamous looks like tiles when you look from the top down. This is actually looking at frog skin. Frog skin is simple squamous epithelium. So here's a few tile-like squamous cells. Sometimes simple squamous epithelium gets special names. Endothelium is the simple squamous epithelium that lines the blood vessels, lymphatic vessels, and the heart. Mesothelium is the simple squamous epithelium that lines serous membranes. Now here I'm showing you simple cuboidal and simple columnar epithelium so that you can compare them side by side. Do you see how the columnar is longer? So here's a few cuboidal cells. Over here you can see they're about as wide as they are tall. And the nucleus sits right in the middle. Here are a few columnar cells. The nucleus a little closer to the basal surface. These two types of epithelium are good at secreting substances, like the simple cuboidal, which you find in glands, and they're good at absorbing substances, like the simple columnar in your digestive tract. Now there are a couple of wacky epithelium types that I want you to take a look at. The first is pseudostratified columnar epithelium. Pseudo means fake. And this is called pseudostratified because it looks like stratified columnar epithelium, but it's really just a single layer of cells. Believe it or not, all of the layers, all of the cells in this epithelium actually touch the basement membrane. So your basement membrane is kind of along there. And it's really hard to see, but if you could look at this three-dimensionally, you'd see that all of these cells do have some attachment to the basement membrane. It's just that their nuclei are at all different levels that makes it look like a stratified tissue. It's very difficult to tell the difference. You just have to know what tissue you're looking at and know that it's supposed to be pseudostratified. This tissue is primarily found in the respiratory tract, where it has cilia along the surface along the apical surface in order to move mucus along. The second type of wacky epithelium that I want you to look at is the transitional epithelium. Transitional epithelium is characteristic of the urinary system. You find it in the bladder and in the ureters. The top layer of cells in this epithelium change shape. When the organ is relaxed, empty, the top layer of cells are large and pillowy, I think they look a little bit like marshmallows. But when the organ fills with urine, the epithelium stretches and the top layer of cells flatten out, looking almost more squamous-like. Alright, this ends our brief tour of epithelial histology. Remember, this is just a beginning, and you really need to spend some time with these slides in lab to familiarize yourselves with the different appearances of these types of tissue. For further reading, I have some references here of places to look in your textbook and in your lab manual. These will give you both more images of tissue to look at and a little bit more background information.